Right, I'm going to quickly go through the phases of the cell cycle and the stages of mitosis. When we're talking about mitosis, the cell is dividing for either growth or repair. It's making an exact copy of itself. When a cell is going about its life, it exists as part of a cell cycle. This is the set of stages from the first division to the time that it divides, if it does divide. There are two major stages of the cell cycle: interphase, where the cell spends most of its time, and mitosis, where the nucleus divides. Within the cell cycle, certain cells do not actually ever divide. They hang out in the G0 stage, like your muscles and neurons. But other cells that actively divide go through this portion of interphase, where they grow, synthesize DNA, and then mitosis, where the nucleus actually divides and we end up with two new cells. During the cell cycle, there are these checkpoints where the cell can stop and make sure everything is where it's supposed to be, make sure everything is lined up, make sure we have enough DNA. Those checkpoints are controlled by signals and cyclins. Again, interphase is where the cell spends most of its time. It's performing its usual functions, but it's going through those G1, S, and G2 phases. The G1 phase is the gap phase, where we're doubling our organelles and growing. The S phase is synthesis, where DNA actually replicates. In the G2 phase, or gap 2 phase, we're synthesizing proteins and materials necessary for that division. When we're talking about division, not only are we talking about the cytoplasm, but the chromosomes. Mitosis is actual nuclear division. So you have 46 copies in your cells, 46 chromosomes, 23 pair. Before mitosis, those chromosomes duplicate. So we see them in this X shape, where we have sister chromatids attached at a centromere. During mitosis, that centromere dissolves and those sisters separate. It allows you to have a cell, and when you divide, end up with two exact copies. Because in mitosis, you don't want any differences, you want exact copies. So mitosis is nuclear division. Cytokinesis is the last portion where we actually divide the cytoplasm. I'm going to skip through a lot of this to get to the stages of mitosis. So in mitosis, we have four major phases: prophase, metaphase, anaphase, and telophase. Really, we should include five. Prometaphase is its own phase in its right. And we finish up with cytokinesis, where the cytoplasm actually divides. In prophase, our DNA has duplicated and we have condensed into these X-shaped chromosomes. The nuclear envelope begins to divide or disappear. Dissolve is the word I meant to say. And these spindles begin to assemble out of the centrioles. They look like little stars or asters. They're just microtubules that are stretching out toward the chromosomes. In prometaphase, on either side of the chromosome, kinetochores form, which are just special proteins that act like magnets that attract the microtubules so that the centrioles can essentially line every chromosome up, make sure it's all there, and then pull into the individual separated cells. In metaphase, this phase you might recall might be the easiest to remember if you think metaphase middle, using the M to help you there, metaphase middle. All of your chromosomes have lined up along the metaphase plate in the middle of the cell. In anaphase, the microtubules are pulling your chromosomes apart. The centromere has dissolved, so those sister chromatids actually separate. They're now called daughter chromosomes and will end up on either side or either pole of the cell. In telophase, we're almost done dividing. Our chromosomes have separated, and so we reform that nuclear envelope. To divide the cell completely, we have to go through cytokinesis, the division of the cytoplasm. In animal cells, a cleavage furrow forms where these proteins squeeze the middle of the cell, growing tighter and tighter like squeezing a lasso, until that ring, that contractile ring of proteins, squeezes the cytoplasm in two. In plant cells, Not only do we have to divide the nucleus and the chromosomes, but we have to reform a cell wall. And so a cell plate forms. Vesicles bring material that eventually hardens and forms a new cell plate in between the cells. So for lecture and lab, you'll need to recall the names of the phases, what occurs in each, and then know the difference between plant and animal cells.